maintain the existence of species, it is essential for mature and fertile adults to produce offsprings. We also know that it is through reproduction that genetic information is passed on from one generation to another. During reproduction, haploid cells called gametes are produced by mature adults. If we talk of humans, the sperms are produced by the testes of males, whereas ova or the female gametes are produced in the ovarian follicles of the females. Sperms and ova are haploid cells. Fusion of sperm and ovum results in the formation of a diploid zygote which develops into a fetus.
ਵਿਦਿਆਰਥੀਓ, ਅੱਜ ਦੇ ਵਿਸ਼ੇ ਬਾਰੇ ਤਾਂ ਤੁਸੀਂ ਸਮਝ ਹੀ ਗਏ ਹੋਵੋਗੇ। ਅੱਜ ਅਸੀਂ ਰੀਪਰੋਡਕਸ਼ਨ ਲਈ ਮਨੁੱਖਾਂ ਵਿੱਚ ਜ਼ਿੰਮੇਵਾਰ ਇੱਕ ਬਹੁਤ ਹੀ ਮਹੱਤਵਪੂਰਨ ਪ੍ਰੋਸੈਸ ਫਰਟੀਲਾਈਜ਼ੇਸ਼ਨ ਬਾਰੇ ਗੱਲ ਕਰਾਂਗੇ। You know that sexual reproduction involves the formation of an individual after the union of two gametes. Fertilization ਲਈ sperms ਦਾ ovum ਕੋਲ ਪਹੁੰਚਣਾ ਹੀ ਕਾਫੀ ਨਹੀਂ ਹੈ। ਸਪਰਮਸ ਵਿੱਚ ਕੁਝ ਤਬਦੀਲੀਆਂ ਦਾ ਹੋਣਾ ਵੀ ਬਹੁਤ ਜ਼ਰੂਰੀ ਹੈ। These changes are collectively known as capacitation. ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ successful fertilization ਲਈ capacitation ਬਹੁਤ ਜ਼ਰੂਰੀ ਹੈ। Capacitation ਦੇ ਦੌਰਾਨ sperms ਦੇ acrosomal region ਉੱਤੇ present glycoprotein coating ਉਤਰ ਜਾਂਦੀ ਹੈ। Decapacitation factors dilute ਹੋ ਜਾਂਦੇ ਹਨ। Calcium ions sperms ਵਿੱਚ ਦਾਖਲ ਹੁੰਦੇ ਹਨ ਅਤੇ ਉਨ੍ਹਾਂ ਦੀ ਮੂਵਮੈਂਟ ਨੂੰ ਤੇਜ਼ ਕਰਨ ਵਿੱਚ ਸਹਾਇਕ ਹੁੰਦੇ ਹਨ। Sperms that have undergone capacitation are said to become hyperactivated. Activated sperms ਓਵਮ ਦੇ ਬਾਹਰ ਆ ਕੇ ਲੱਗ ਜਾਂਦੇ ਹਨ ਤੇ ਅੰਦਰ ਦਾਖਲ ਹੋਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰਦੇ ਹਨ। ਚਲੋ ਇਸ ਨੂੰ ਵਿਜ਼ੂਅਲ ਰਾਹੀਂ ਸਮਝੀਏ।
Fertilization. The fimbriated end of the fallopian tube receives secondary oocyte. These oocytes can live for about 72 hours but can be fertilized only during the first 24 hours or so. The egg is covered by a layer of follicle cells known as corona radiata. This is a multicellular layer with intercellular matrix of proteins, hyaluronic acid, and carbohydrates. The process of fertilization involves a sperm fusing with an ovum after ejaculation during sexual intercourse. The union of an egg and sperm usually occurs in the ampulla region of the fallopian tube. Spermatozoa are not able to fertilize the oocyte immediately upon arrival. During their journey, sperms also undergo capacitation. To reach the ovum, each tiny sperm must travel about 30 to 40 centimeters of the female reproductive tract. This movement is possible due to the flagellar activity of the sperms as well as contraction of the uterus and fallopian tubes. Efforts are made by millions of sperms, but only a few happen to complete their journey. Although many sperms contribute enzymes to disrupt the follicular cells, but only one sperm can make entry into the ovum. The site where fertilization usually takes place is the ampullary isthmic junction of the fallopian tube.
Students, You know the sperm and the ovum are two haploid cells, and it is the process of fertilization that restores the diploid number of chromosomes. The union of a sperm and egg is an extraordinary cell fusion that gives rise to the usually diploid number of chromosomes and triggers a very sophisticated developmental process. Actually, as the sperm head reaches the zona pellucida, compatibility reaction occurs, which results in emptying of acrosome and degeneration of zona pellucida. Receptor proteins of zona pellucida have been identified as ZP1, ZP2, ZP3. ਤੁਸੀਂ ਜਾਣਿਆ ਕਿ ਸਪਰਮ ਨੂੰ ਓਵਮ ਵਿੱਚ ਐਂਟਰ ਕਰਨ ਤੋਂ ਪਹਿਲਾਂ ਕੁਝ ਬਦਲਣਾ ਪੈਂਦਾ ਹੈ, ਜਿਸ ਨੂੰ ਅਸੀਂ ਕਪੈਸੀਟੇਸ਼ਨ ਆਫ ਸਪਰਮਸ ਕਹਿੰਦੇ ਹਾਂ। ਇਹ ਉਹ ਫਿਨੋਮੀਨਨ ਹੈ ਜਿਸ ਨਾਲ ਮੈਮਲਸ ਦੇ ਸਪਰਮਸ ਐਕਟੀਵੇਟ ਹੁੰਦੇ ਹਨ। ਕਪੈਸੀਟੇਸ਼ਨ ਟੇਕਸ ਅਬਾਊਟ 5 to 6 hours। ਇਸ ਤੋਂ ਬਾਅਦ acrosomal reaction ਹੁੰਦੀ ਹੈ। As soon as sperms come in contact with the surface of the egg, acrosome, ਜੋ ਕਿ sperm ਦਾ ਸਭ ਤੋਂ ਅਗਲਾ ਭਾਗ ਹੁੰਦਾ ਹੈ, ਹਾਈਡਰੋਲਿਟਿਕ ਐਂਜ਼ਾਈਮਸ ਛੱਡਦਾ ਹੈ, ਜੋ ਕਿ ਇੱਥੇ ਮੁੱਖ ਤੌਰ ਤੇ ਕੋਰੋਨਾ ਪੈਨੀਟ੍ਰੇਟਿੰਗ ਐਂਜ਼ਾਈਮਸ ਹੁੰਦੇ ਹਨ। ਇਹ ਐਂਜ਼ਾਈਮਸ ਕੋਰੋਨਾ ਸੈਲਸ ਨੂੰ ਆਪਸ ਵਿੱਚ ਚਿਪਕਾਉਣ ਵਾਲੇ ਸਬਸਟੈਂਸ ਨੂੰ ਡਿਸੋਲਵ ਕਰ ਦਿੰਦੇ ਹਨ। ਇਸ ਕਰਕੇ ਕੋਰੋਨਾ ਸੈਲਸ ਵੱਖਰੇ ਹੋ ਜਾਂਦੇ ਹਨ। Certain enzymes which help in degenerating corona cells are also released. ਚਲੋ ਇਸ ਨੂੰ ਵਿਜ਼ੂਅਲ ਰਾਹੀਂ ਸਮਝੀਏ।
Once the sperm has cut through this layer of corona radiata using the corona penetrating enzymes, it encounters the zona pellucida just where it is needed. Means, hyperactivated sperms reach close to the ovum, where binding of the sperm to the zona pellucida of ovum takes place. It is a receptor-ligand interaction with a high degree of species specificity, or we may say compatibility. Binding is mediated by the ligand ZP3, a zona protein, and receptors on the zona pellucida. During continued acrosomal reaction, the release of more hydrolytic enzymes act as an enzymatic drill to get through the zona pellucida. The sperm remains attached to the egg and subsequently penetrates the zona pellucida with the aid of an acrosomal protease, zona lysin. Release of hyaluronidase then takes place from the sperm head, which destroys an intercellular material, hyaluronic acid, present between the layers. Now, the cortical reaction takes place, which leads to a massive exocytosis of cortical granules. The cortical granules contain certain proteases that alter the structure of the zona pellucida. Sperm receptors of the zona pellucida are destroyed, and it becomes hardened to prevent more sperms from entering. The egg is said to be activated after fusing with a single sperm cell, thereby changing its cell membrane to prevent fusion with other sperms. Therefore, the sperms that have not yet bound to the zona pellucida will no longer be able to bind, thereby preventing polyspermy.
ਵਿਦਿਆਰਥੀਓ, ਇਹ ਸਭ ਬਹੁਤ ਫਾਸਟ ਹੁੰਦਾ ਹੈ। Within moments, enzymes released from the egg cytosol act on the zona pellucida, making it impermeable to the other sperms. इस करके पोलीस्पर्मी ते कंट्रोल रहंदा है. This is what happens within the fallopian tube once the sperms reach the ovum. You have seen many sperms competing with each other, but only one happens to make it. Now fusion of their respective membranes allows the entire content of the sperm to be drawn into the cytosol of the egg. You know, even though the sperm's mitochondria enter the egg, they are almost always destroyed and do not contribute their genes to the embryo. So ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ ਕਿ mitochondrial DNA is almost always inherited from the mother only. ਚਲੋ ਹੁਣ ਅੱਗੇ ਵਧਦੇ ਹਾਂ ਅਤੇ ਵੇਖਦੇ ਹਾਂ ਕਿ ਜਦੋਂ ਸਪਰਮ ਅਤੇ ਓਵਮ ਮਿਲਦੇ ਹਨ ਤਾਂ ਓਵਮ ਦੇ ਨਿਊਕਲੀਅਸ ਦੇ ਅੰਦਰ ਕੀ ਹੁੰਦਾ ਹੈ. The nucleus of the successful sperm enlarges into the male pronucleus. At the same time, the secondary oocyte completes meiosis II, forming a second polar body and the female pronucleus. आओ इस ਨੂੰ ਐਨੀਮੇਸ਼ਨ ਦੀ ਮਦਦ ਨਾਲ ਸਮਝਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰਦੇ ਹਾਂ.
Fertilization. This process ultimately leads to the formation of a diploid cell called a zygote. Let us see how it is formed. During the fusion of a sperm with an ovum, the sperm head gets incorporated into the egg cytoplasm. The entry of the sperm stimulates the secondary oocyte to resume the suspended meiosis II. This produces a haploid mature ovum and a second polar body. After oocyte activation and completion of meiosis, the nuclear material remaining within the ovum reorganizes as the female pronucleus. The male pronucleus migrates towards the center of the cell, and the two pronuclei fuse in a process called amphimixis. So we can say that amphimixis is a process through which mixing up of chromosomes of a sperm and an ovum takes place. It is also known as karyogamy. 23 chromosomes from the male pronucleus and 23 chromosomes from the female pronucleus fuse to form the human zygote as a result of amphimixis. Thus, a zygote containing a diploid number of chromosomes is formed.
Students, ਅਸੀਂ fertilization ਬਾਰੇ ਸਮਝ ਗਏ ਹਾਂ. Fertilization ਦੇ ਦੌਰਾਨ ਕੀ ਹੁੰਦਾ ਹੈ ਉਸ ਨੂੰ ਅਸੀਂ ਦੁਹਰਾਈਏ ਤਾਂ ਮੁੱਖ ਪੁਆਇੰਟਸ ਇਹ ਹੋਣਗੇ. Restoration of a diploid number of chromosomes. Determination of the sex of the baby. One has to remember that the sex of the baby has been decided at this stage itself. As you know that the sex chromosomes in the females are XX and that in the male are XY. Therefore, the haploid gametes mean the ova produced by the female have sex chromosome X, whereas the male gametes means sperms will either have sex chromosome X or sex chromosome Y. Or we may say 50% of the sperms carry X chromosome, while the other 50% carry chromosome Y. After fusion of the male and female gametes, the zygote would carry either XX or XY, depending on the sperm that fertilizes the ovum. The zygote formed by fusing with the sperm carrying X chromosome will have XX chromosomes and develops into a female baby. But if a sperm carrying Y chromosome fertilizes the ovum, the zygote would have XY chromosomes and develops into a male baby. As the sperm and ovum have been fused, so scientifically it is correct to say that the sex of the baby is determined after this stage. Initiation of cleavage is the next stage of the process, or we may say that all this is the outcome of fertilization. Fertilization indicates the beginning of one of the most remarkable phenomena in the body, that is, the process of embryogenesis. Embryogenesis is a process through which the zygote develops into a new individual in the form of embryo. As I told you, a fertilized ovum is known as a zygote. Now, this zygote undergoes rapid mitotic divisions by a process known as cleavage. Actually, no significant growth takes place during cleavage. It produces a cluster of cells equal to the size of the original zygote. The cells produced as a result of cleavage are called blastomeres, and the compact mass formed is called morula. The process of cleavage ends with the formation of the blastula. Cellular differentiation occurs during further cleavage that leads to the development of an embryo. Basically, cleavage generates a multicellular system essential for further differentiation and morphogenesis. चलो इसनू भी विजुअल राही समझिए.
Cleavage. Fertilization initiates cleavage, and once the zygote reaches the two-cell stage, it undergoes a series of rapid mitotic divisions, thus increasing the number of cells. The first cleavage divides the cell into two, and the second cleavage produces the four-cell stage, and so on. Approximately 3 days after fertilization, cells of the compact embryo divide to form a 16-cell stage known as morula. The solid morula then develops a fluid-filled cavity, and this way a blastula is formed. The fluid-filled cavity of the blastula is called the blastocele. This cavity is surrounded by a layer of cells called blastomeres. When the cavity enlarges, the form is known as a blastocyst. Cells of blastocyst are distributed into two groups. One composed of an outer envelope of cells, the trophoblast, and the other set of cells form the embryoblast. This side of the blastocyst to which inner cell mass embryoblast is attached is called the embryonic or animal pole. This stage in the developing embryo is reached after 4 to 6 days and lasts approximately until it gets implanted in the uterus. So we can say that till this stage the embryo is known as blastocyst.
The blastocyst remains free within the uterine cavity for 2 to 4 days before it attaches to the uterine wall. इस स्टेज ते इसनू प्री-implantation embryo भी कहते हैं. Each cell of the pre-implantation embryo is totipotent. Totipotent ਦਾ ਮਤਲਬ ਤਾਂ ਤੁਸੀਂ ਜਾਣਦੇ ਹੀ ਹੋਵੋਗੇ। ਇਸ ਦਾ ਮਤਲਬ ਹੈ each cell has the potential to form all the different cell types in the developing embryo. Cells ਦੀ ਇਸ tendency ਨੂੰ totipotency ਕਿਹਾ ਜਾਂਦਾ ਹੈ। Cleavage process ਦੀਆਂ ਦੋ ਮਹੱਤਵਪੂਰਨ stages morula ਅਤੇ blastula ਹਨ। ਚਲੋ ਇਹਨਾਂ ਦੋਹਾਂ ਨੂੰ ਯਾਦ ਕਰਕੇ ਇਹਨਾਂ ਵਿੱਚ ਫਰਕ ਨੂੰ ਵੇਖਦੇ ਹਾਂ। Morula is a solid ball-like structure, whereas blastula is a hollow structure with a cavity in it. Morula is made up of similar kind of cells. On the other hand, blastula is made up of two different layers of cells, trophoblast and embryoblast. The next step is implantation. Implantation is the attachment of the blastocyst to the uterine wall. For the implantation of the blastocyst, trophoblastic cells over the embryoblast pole begin to attach themselves to the endometrium, and the blastocyst continues to invade the uterine wall. By this time, trophoblastic cells start secreting the human chorionic gonadotropin hormone, means HCG. The HCG maintains the corpus luteum and causes more secretion of progesterone. This hormone maintains the endometrium of the uterus and causes it to grow during pregnancy. So students, now the blastocyst is ready to get embedded in the uterine wall. The blastocyst fully attaches itself to the endometrium through a very complicated process. By the 11th or 12th day of development, when the blastocyst gets completely embedded in the endometrial stroma, it starts producing hormones which can be detected in the urine. Means, conceiving can be detected medically through a small urine test. This is the event that almost all physicians define as the start of pregnancy. ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਇੰਨਾ ਹੀ ਕਾਫੀ ਹੈ। ਚਲੋ ਇਸ ਨੂੰ ਦੁਹਰਾ ਲਈਏ। Fertilization is the union of an egg and sperm, usually occurring in the ampulla region of the fallopian tube. Freshly ejaculated sperms are unable to fertilize. Rather, they must first undergo a series of changes, collectively known as capacitation. Fertilization begins with the binding of a sperm head to the outer coating of the egg called the zona pellucida. Exocytosis of the acrosome at the tip of the sperm head releases enzymes that digest a path through the zona pellucida and enable the sperm head to bind to the plasma membrane of the egg. Within minutes, enzymes released from the egg cytosol in the form of cortical granules act on the zona pellucida, making it impermeable to other sperms, thus avoiding polyspermy. Soon the nucleus of the successful sperm enlarges into the male pronucleus. At the same time, the egg, means secondary oocyte, completes meiosis too, forming a second polar body and the female pronucleus. The male and female pronuclei fuse to form the fertilized egg or zygote. Actual development begins even when the fertilized egg is still within the fallopian tube. repeated mitotic divisions produce a solid ball of cells called a morula and then a blastula. Further mitosis and some migration of cells converts this into a hollow ball of cells called the blastocyst. Approximately 1 week after fertilization, the blastocyst gets embedded in the thickened wall of the uterus, a process called implantation. And with this, a pregnancy is established. So students, ਮੈਨੂੰ ਲੱਗਦਾ ਹੈ ਕਿ ਅੱਜ ਦੀ ਕਲਾਸ ਲਈ ਇੰਨਾ ਹੀ ਕਾਫੀ ਹੈ। ਅਸੀਂ ਆਪਣੇ ਪਾਠ ਨੂੰ ਸਿੰਗਲ ਸੈਲਡ ਹੈਪਲੋਇਡ ਗਮੀਟਸ ਨਾਲ ਸ਼ੁਰੂ ਕੀਤਾ ਅਤੇ ਵੇਖਿਆ ਕਿ ਸਕਸੈਸਫੁਲ ਫਰਟੀਲਾਈਜ਼ੇਸ਼ਨ ਤੋਂ ਬਾਅਦ ਕਿਵੇਂ ਸਿੰਗਲ ਸੈਲਡ ਹੈਪਲੋਇਡ ਗਮੀਟਸ ਆਪਸ ਵਿੱਚ ਮਿਲ ਕੇ ਇੱਕ ਰੈਕਗਨਾਈਜ਼ੇਬਲ ਫਾਰਮ, ਜਿਸ ਨੂੰ ਜ਼ਾਈਗੋਟ ਕਹਿੰਦੇ ਹਨ, ਵਿੱਚ ਬਦਲ ਜਾਂਦਾ ਹੈ। आओ हुण कुछ सवाल पुछ के ये पता करते हाँ कि तुहानू अज दी इस क्लास विच किन्ना कुछ समझ आया है। फर्स्ट क्वेश्चन, what do you mean by hyperactivated sperms? Answer, sperms that have undergone capacitation are said to become hyperactivated. Next question, what is capacitation with reference to sperms? Answer, changes in mammalian sperms which prepare them to fertilize an ovum is called capacitation. Next question, hyaluronidase is present in ovary, ovum, sperm or blood? Answer, yes, you are right. Hyaluronidase is present in sperm. Its release takes place from the sperm head, which destroys an intercellular material, hyaluronic acid, present between the layers of the ovum. The next question is, which of the following statements is correct? During cleavage, size of embryo increases, or size of cells decreases, or size of cells increases, or size of embryo decreases? Answer, during cleavage, size of cells decreases. The next question is, identify the correct sequence. Following are the options. First, fertilization, zygote, morula, blastula. Second option, zygote, fertilization, morula, blastula. Third option, fertilization, blastula, morula, zygote. Answer, the correct sequence is fertilization, zygote, morula and then blastula. My next question is, name the hormone present in urine that is used in the pregnancy test. Answer, presence of human chorionic gonadotropin hormone means HCG is detected during a pregnancy test. ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ ਅੱਜ ਦਾ ਇਹ ਪਾਠ ਚੰਗੀ ਤਰ੍ਹਾਂ ਸਮਝ ਆ ਗਿਆ ਹੋਵੇਗਾ। ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਧਿਆਨ ਦੇਣ ਲਈ ਸ਼ੁਕਰੀਆ। ਅਗਲੀ ਵਾਰ ਇੱਕ ਨਵੇਂ ਟੌਪਿਕ ਨਾਲ ਫਿਰ ਮਿਲਾਂਗੇ। ਗੁੱਡ ਬਾਏ।